Very good morning to one and all present here. My name is Dr. Vivekanandhan, Head and Assistant Professor, Department of Microbiology, KSR College of Arts and Science for Women. Today our topic is Microbial Physiology and Metabolism. First of all, you can very well about what is this culture. The microbiological culture or microbial culture, it is a method of multiplying microbial organism, then reproduce in predetermined culture medium under controlled our laboratory conditions. Microbial cultures are foundational and basic diagnostic methods using the research tools in our molecular biology. What are the contents of our microbial metabolism? The first one is batch culture. Then what are the different 6 phases of cultures methods? What are the continuous culture? Then what is the synchronous culture? What is the main application of our culture system? What are the environmental factors on the growth system? Our batch culture means generally the closed culture system that contains limited amounts of nutrients we can apply to this system. In this bacterial batch culture, we can sterile growth mediums inoculated in the appropriate microorganism, and cultures proceed with the addition of any fresh growth medium. Then after inoculating the medium and start measuring the biomass at a different time intervals. You may fix in six different phases under these conditions. Usually for these 6 phases are observed. The first one is the lag phase, then accelerate phase, then expositional phase, then declared growth phase, then stationary phase, the finally death phase. The lag phase, this time between the inoculation of and beginning multiplication is known as a lag phase. The organism is starting multiplication process. Then lag phase is the initial phase, the period of cultivation during which the changes of the cell numbers is 0 or negative. During this phase, there is an increase in the sizes of bacteria, but not applicable increase the number of bacteria. Only for the size it will be increasing. Even though the cell members does not increases, the cells grows during during the sizes of period. As the duration it may differ in 1 hour to the several days. Then acceleration phase, at the end of this phase, when the growth is begin, the division rate is increasing gradually. After reactions, the maximum value at its called acceleration phases. Then what are the log and expositional phase? This phase is characterized by rapid expositional growth. The organism range is 1 to 2, 2 to 4 and 4 to 8. This bacterial populations doubles during this every generation. The cell divisions occur in the different phases. The bacterial cells are small and uniformly stained. Then the 4th phase, declaration phase or growth phase. The end of the expositional phase occurs when the nutrients depleted or toxic metabolites will be accumulated in this our system. During this phase, growth rate will be decreasing. The toxins may be affecting our growth of the organisms. The 5th one is the stationary phases. During this phases, the cultures it is a greatest population and density. However, death rate of bacteria exceeds the rate of replication of bacteria. And the endospores start forming during the stages. The endospore is forming the internal spores will be following our bacterial culture. The bacteria become a gram variable and is showing the irregular staining culture. The many bacteria they start producing exotoxin materials. What are the factors will be affecting the phases? The nutrition limitation. The nutrients, particular nutrients one will be increasing the growth of range of bacteria. This is called nutrient limitation. This is essential nutrient is severely depleted and population growth will be slow. Then aerobic organism are often or limited of oxygen as O2 availability. The cells beneath the surface will not able to grow unless the culture is shaken or aerated is another way. Due to the accumulations of toxic waste products will be accumulated. Then the final phase is death phase. During this phase, this is the decline phase started due to the accumulation of toxic products and the autolytic enzymes. The autolyting with the self-lighting. The self-toxic materials the lighting the bacterial cells. It's called the autolytic enzymes. And the explosion of the nutrients materials. The excess amount of nutrients will be debited, will be affected over the death phase. Some cells assumes that the various shapes becomes a long and the filamentous or branching system. Some developments of without cell wall are referred to the protoplast or spheroplast material. Even then, bacterial cells were transferred from the fresh medium. No cellular growth was observed because loss of viability often to the not accompanied with the loss in the total number of cells. Thank you for listening this our video lecture. Thank you.